Hello and welcome to episode number 19 of Gowing Life's Longevity Weekly. I'm your host Chris Cohen, and this is the show where I bring you all the news and updates from the world of health, wellness and longevity. And today we're going to be talking about a series of articles written by Alex on the hallmarks of aging, my chat with Nikhil Yadala, CEO and co-founder of Healome, and also part 2 of studying neurological disease with induced pluripotent stem cells. So without further ado, let's get right into it. Now firstly, as a legendary Professor Richard Feynman once said, there is nothing in biology yet found that indicates the inevitability of death. This suggests to me that it is not at all inevitable. As far as we know, this statement still holds true. But in order to materialise the implication of Professor Feynman's statement, that we will, in fact, one day cure all disease, we must understand the fundamental changes that underlie the biological process of ageing. Now, ageing is a collection of interconnected problems, and is unlikely to be fully explained by a single factor. The biological processes which seem to universally drive or are associated with ageing were coined the hallmarks of ageing by a seminal paper published in 2013. The hallmarks include genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. Now, by breaking ageing down into its constituent components, we may study which ones contribute the most to the ageing process as a whole, and we may begin to search for links between all of these hallmarks that may lead us to targets for reversing the ageing process entirely. Now, by all means, this is no easy task, due to the incredible complexity of these processes. Now, we consider these as the hallmarks of ageing because they are the common denominators of ageing, not only amongst humans, but also throughout different species. Furthermore, at least one or two of them, and in many cases most of them, are found in all chronic diseases of ageing. And also the study of genetic mutations affecting these hallmarks, both in animals and humans, suggests that causal relationships exist between them and the ageing process. Now, over his series of articles, Alex discusses each hallmark of ageing in detail, including what exactly they are, how they change during ageing, and how we might be able to reverse them in the future. So hopefully by the end of this series you will have a wider understanding of what actually makes us age. And secondly, earlier this week I had the absolute pleasure of being joined by Nikhil Yadala, the CEO and co-founder of Healome. Now, Healome is a service which allows its users to store all of their digital medical information. Using artificial intelligence and machine learning techniques, Nikhil and his team analyse your data, enabling healthcare professionals to provide valuable insight to enhance you and your family's health. Now, here is just a short snippet of our talk. So what kind of problem is Healome actually attempting to solve?
First and foremost, Healome is a platform that lets users organize their healthcare information. In most of the developing countries, there is no concept of electronic healthcare record or EMR system. People carry the healthcare prescriptions in physical hard copies in files. So if you go to a doctor, writes something with a pen on a piece of paper and gives it to you, and by the time someone reaches the age of 40, they just have a bunch of papers lying around in order to show their healthcare information to someone. Like if somebody falls unconscious on road and if he's admitted to an ICU ward, there is literally no way for the doctor to learn about their previous health condition, to get clues as to why have they fallen unconscious. So through Healome, you can look at all of your information through your mobile application and search through them and track various records. So that's one thing. And secondly, by proactively looking at various signals in the blood biomarkers and other specific health information that we get from the users, so we prune for those signals to detect abnormal patterns at a very early stage so that we can hopefully change the trajectory of the disease progression.
So thanks again to Nikhil for giving us his time, and you can go and check out the full talk on our YouTube page. So last week I talked about part 1 of an article series which looked at the global impact of neurological disease, the challenges in trying to study them, and the introduction of induced pluripotent stem cells to help model them. But what exactly are induced pluripotent stem cells, and why are they so important? In 2007, Dr. Yamanaka and his team in Japan injected viral transport particles containing 4 proteins known as the Yamanaka factors into adult cells. These Yamanaka factors were found to drive the process of turning adult cells back into a pluripotent, undifferentiated state. One of the most amazing things about this process is that it can be done using almost any type of cells. Induced pluripotent stem cells also have the ability to turn back into any cell type. This means that scientists can take some very easily accessible cells, skin cells for example, and then using these Yamanaka factors turn them back into stem cells. These pluripotent stem cells, which can then be differentiated into brain cells or any other cell type which is difficult to obtain. These cells can then be used to simulate a certain disease, to determine its unique mechanisms, or be used as a dummy for running potential drug or interventions on, to see how they respond. Now this is known as a disease model. Another crucial benefit of using these induced pluripotent stem cells to build disease models is that they can be derived from the cells of the individuals with a specific disease. This means that the resulting disease model contains the exact genetic defect and identical genetic background of the individual the cells have come from, allowing for a model that far more accurately represents a specific disease. Therefore, patient samples can be used to develop induced pluripotent stem cell lines that host pathogenic mutations. Subsequently, using gene editing techniques such as CRISPR-Cas9, iPSC lines can be genetically modified to edit a particular genetic loci and ascertain the relative contribution of a specific mutation to a disease characteristic. This allows the examination of the disease in greater detail and also enhances the capacity to correct the desired cells for the production of novel cellular therapies. Since this important discovery by Yamanaka and his team, developments to efficiently produce iPSCs from somatic cells have continued at a steady pace. Now keep an eye out for part 3 where we'll be talking about how to use iPSCs to build the ultimate disease model, artificial brains. And that is it for another episode of Longevity Weekly, so thank you so much for joining again. If you'd like to find out more, then I've left all the links in the description below. Also go and check out the Gowing Life website, where you can check out a whole host of other articles, including some of the Hallmarks of Ageing articles that I mentioned earlier, and also an article about the TRIM trial, the first study to reverse human ageing. So go and check it out. I look forward to seeing you again, and have a great week. Thank you very much.